All right, thank you, Stella. Um, can everyone see my screen? And do you see my notes, or is it just the title page?
I just see your title page. Yeah.
Okay, perfect. All right, so yeah, thank you for that introduction, Stella. And yeah, today I'm going to be presenting some of the work from my postdoctoral work, so my postdoctoral fellowship and my PhD work. So just a brief about me. I'm a dad to two kids. I have a 1-year-old daughter named Maisie and a 4-year-old son named Wesley. I'm from Florenceville. That's where I was born and raised. I attended UMB Fredericton from 2009 to 2013, where I completed my honors degree in psychology. And this paper is a direct result of my honors degree. As you can see, it was in social psychology. And as you can see, I completed my degree in 2013, and this paper came out in 2019. Believe it or not, that's not the largest gap between when a project was done and when it was published. Uh, I had a manuscript set on my PhD supervisor's desk for 8 years. So yeah, as Stella mentioned, I completed my PhD at the University of Saskatchewan, completed in 2019. That was in neuroscience. I did a postdoctoral fellowship at Memorial University from 2019 to 2022. And then I did some work with NBCC as a research associate in health and from 2022 to 2025. And I just started with Horizon Health Research Services in November of this past year. So yeah, it's still pretty new, but yeah, definitely enjoying myself so far. So I think the story of my PhD and postdoc is really the story of what the hell happened to my hair. Um, I'm still looking for it. It's gone. If you ever see it, please let me know. So major depressive disorder is is a serious psychiatric disorder which affects approximately 350 million people worldwide. And its diagnosis is informed by the presence of first cardinal symptoms. So cardinal symptoms there's two of them. The first being anhedonia. So anhedonia refers to the loss of pleasure in what was previously a pleasurable activity for an individual. So for example, if before depression, say someone was an avid runner and reader, after depression, if they had anhedonia, they're most likely not going to be interested in running and reading anymore. An additional cardinal symptom is lowered mood. And so they must have one of two of these cardinal symptoms. In addition to these cardinal symptoms, there must be the presence of some additional symptoms, which include insomnia or hypersomnia, weight gain or weight loss, feelings of worthlessness and guilt, and psychomotor retardation or agitation. As you can see from this, it's quite a spectrum because you have almost completely opposite symptoms that patients can have. And there's been lots of commentary on that in the literature. One thing I'd like to bring to your attention is the kindling hypothesis. So the kindling hypothesis is similar to an epilepsy, where basically once you have an episode of depression, your brain becomes primed and it's a bit more easier to subsequently have more depressive episodes. And that was a really interesting paper that my lab had published, which was looking at trying to model this kindling hypothesis in a rodent model of depression. In terms of pharmacology, there's 4 medication classes. The first are the MAOIs, or the monoamine oxidase inhibitors. These are the first class of medications developed, and they were actually serendipitously discovered. They're not widely used right now, but they do still have some utility in some instances. Oops, sorry. The second class of antidepressant medications are the TCAs, or the tricyclic antidepressants. These are characterized by the 3-ring structure, and they represent a pretty marked improvement over the MAOIs, but there are some side effects in terms of acetylcholine signaling, although they have been shown to be quite useful in treatment-resistant depression. The third class are the SSRIs or the SRNIs, so selective serotonin reuptake inhibitors or selective norepinephrine reuptake inhibitors. And these are the most widely used today. They have much reduced side effects compared to the other medication classes. And then the fourth class would be atypical antidepressants, which are medications that are used to treat depression, but which were not actually developed originally for the treatment of depression, but which have been observed to have some effects on monoamine signaling. However, there are significant issues with these medications. The first off is effectiveness. So it's been estimated that at the high end, only about 55 to 65% of patients experience true depression remission. So that's indicating that upwards of 35 to 45% of patients are not actually experiencing remission, which is quite troublesome. The second one is all antidepressants suffer from a therapeutic lag. And this refers to the time it takes from when a patient starts taking the medication to when they actually start experiencing an antidepressant effect. And this can take anywhere from 4 to 8 weeks. This is a huge issue seen as how one of the main symptoms of depression is suicidality. So this underscores the need for new candidates not acting on these traditional monoamine-based systems. So the cytokine sickness hypothesis of depression arose from the observation that patients with heightened immune activity frequently display the hallmark features of depression. So for example, lethargy, depressed mood, and anhedonia. And this led to the observation that there are high levels of pro-inflammatory cytokines that are present in depressed patients. So this includes TNF-alpha and interleukin-6 as an example. And pro-inflammatory cytokines, what they do is they kind of promote inflammation, and they promote even greater release of other pro-inflammatory cytokines. And then if you look at inflammatory conditions, we frequently see that they share a high comorbidity with depression. So if you look at rheumatoid arthritis, there's about a 13 to 42% comorbidity rate with depression. And I linked a paper here at the bottom, which showed that if you take depressed patients and you stratify them into levels of what's called hsCRP, so high sensitivity C-reactive protein, it's just a marker of inflammation. What you see in those depressed patients with high levels of hsCRP, then the TNF-alpha antagonist infliximab was highly effective in treating depression in these patients. But in depressed patients with low levels of this hsCRP, so low levels of inflammation, infliximab was not very effective in treating depression. So this brings me to the first paper I want to discuss for my PhD work, which was looking at the antidepressant effects of etanercept, which is a TNF-alpha antagonist in a rodent model of depression. So I'll just briefly describe the model that we use. So the model I used was called the repeated corticosterone, or CORT, injection model of depression. So corticosterone is the primary stress hormone that's released in rodents. The equivalent in humans would be cortisol. And so we do 21 days of injections each day. They get 40 milligrams per kilogram of CORT, or vehicle, or just saline. Then we do the forced swim test on day 22, which I'll explain here in a little bit. Then we did a couple days of top-up injections just to kind of maintain that depressive phenotype. We did some cognitive testing, which I will again explain later. And then we sacrificed our rodents. So the first thing I'm going to point you to is body weights. So we always look at body weights in these studies, and that is because our CORT-treated rats, or our depressed rats, have lower body weights than non-stressed rats. And yeah, again, this is what we see. Our stressed rats weigh less. Depressed rats treated with etanercept do not recover body weights, which is fine. So here I have the results for the forced swim test. So what the forced swim test is it's a very commonly used measure of depressive-like behavior in rodents. And so you have this large tank that's filled with water, and basically you just take the rat, you put them in the tank of water, and you record them for 10 minutes. And we want to see there's a certain set of behaviors that always emerge from rats when they're placed in the forced swim test tank. And these are immobility, struggling, and swimming. So immobility is kind of as the name suggests, the rat just stays immobile in the tank. And what we found is that, as we always see, our depressed rats spent significantly more time immobile in the test, so they gave up faster. And our CORT rats that were treated with etanercept, shown here, right beside the black bar, they spent significantly less time immobile, so they behave like non-stressed rats. Another behavior is struggling. So these are kind of active behaviors using their front paws or their back paws, or all of them in combination to actively try and escape out. And what we see is that our stressed rats spend significantly less time struggling, because again, they're giving up more often. And our CORT rats that are given etanercept are behaving like control rats. They are struggling a lot more. And the last behavior is swimming, which is just kind of, it's like a non-goal oriented behavior where they're just kind of swimming point to point in the tank. So it's an active behavior. And again, we see our stressed rats are spending significantly less time swimming, and our CORT rats treated with etanercept spend significantly more time swimming. So what we see is that etanercept is reversing depressive-like behavior in our depressed rats. We also did some cognitive testing, as I mentioned. So the first one was object location testing. This is a measure of hippocampal functioning. And what you do is you have a box and you have two objects that you place in corners of the box. You place the rats in, they explore, and then that's the training phase. And then 24 hours later, you do a testing phase where you switch the orientations of one of the objects. And what you would expect a rat to do is they're going to spend more time exploring the object in the novel orientation because they're going to recognize this is something novel, this is new, and so they're learning that this has changed. And what we see is that our stressed rats, so our court-treated rats, had a significantly worse memory for this task. They spent more time exploring the object in the familiar orientation. And our court rats that were treated with etanercept are displaying normal object location memory, similar to unstressed rats. The other test we looked at was called the object in place testing. This test is also reliant on the hippocampus, but also has prefrontal cortex involvement. And as you can see in this diagram, what you have is you have 4 objects. You put them in the box for a sample phase for 5 minutes, and then you let them freely explore. 1 hour later, we do a test phase where we switch the orientations of objects on one side of the box. So you can see if you look on the sample phase on the left side, you can see their orientation in the test phase has now switched, whereas the objects on the right side maintain their original orientation. So again, what we see is that our stress rats are spending more time exploring the objects in the familiar orientation, indicating that they have not learned this test, whereas our stress rats treated with etanercept are displaying normal object in place testing memory. So what this is showing is that etanercept is able to reverse depressive-like behavior and depression-induced cognitive deficits. Another thing we wanted to look at was neurogenesis. So neurogenesis is, it's not exclusive, but it's observed in the hippocampus, one area, and it refers to the birth of new neurons. And what we see is that, as usual, it's been shown in the past, neurogenesis is reduced in our stressed rats. They have lower numbers of these complex cells, and they have a lot more of these immature, newly born cells. And our stressed rats that were treated with etanercept have restored neurogenesis. And this is just kind of showing a picture where you can see we're looking at the dentate gyrus here in the hippocampus, and it just shows representative pictures between each of the groups. It's a bit hard to see, though, I imagine. Another thing we wanted to look at was hippocampal renin expression. So renin is an extracellular matrix protein that's involved in functions promoting synaptic plasticity. It's heavily implicated in schizophrenia, Alzheimer's disease, and depression. And what's been shown in the past by it's not my lab anymore, but my previous lab, was that stressed rats have significantly reduced hippocampal renin expression. And what we saw is that our stressed rats treated with etanercept had restored levels of hippocampal renin expression. So why this is important is one of the seminal works from my previous lab looked at the progressive development of depression-like behavior and hippocampal renin expression. And what they showed is that after about 7 days of corticosteroid injections, you start seeing depressive-like behavior. This increases by day 14 and is at its highest by day 21. And what was interesting is that hippocampal renin expression coincides with this appearance of depression-like behavior. So by day 7, you start to see some loss of hippocampal renin expression. By day 14, you see much enhanced loss of hippocampal renin expression. By day 21, you see significant loss of hippocampal renin expression. So this led us to believe that perhaps etanercept is achieving its antidepressant effects through its effects on renin signaling. So this led us to the second paper and the largest study of my PhD, which was looking at the antidepressant properties of renin in this corticosteroid model of depression. And I'm just going to say right now, all I see is my slides. So if anyone has questions, like their hands up, I'm not seeing that. So if anyone has questions, they can just feel free to interrupt me at any time. So what we did in this study is, again, we took rats, we injected them with corticosteroid for 21 days or saline. And then in some rats, they received injections of renin at the same time. So how we did this is we took rats, we put them in a stereotaxic apparatus. So they put their heads in a system, we put them under anesthetic, and then we performed brain surgery. So we would drill a hole into their hippocampus, put a cannula down in there, and then we could infuse renin directly into the hippocampus. So some rats get this on days 7, 14, and 21, that they were called the repeated renin group. And some rats only received renin on day 21, 1 day before we did our forced swim test. And this was called the acute group. And so this really allowed us to see the kind of more chronic effects of renin and acute effects of renin. And yeah, so then we ran them through the forced swim test. We did the object location, so that hippocampal dependent memory test, and then we sacrificed them. And then I'll touch on this later. Another cohort of rats only received Relan on day 21, and they also received an injection of what's called CNQX. And CNQX is a glutamate receptor antagonist. And then we ran them through the forced swim test. So what was really interesting and exciting was that what we found is that if we're looking at the forced swim test, again, we want to look at immobility, so the total time the rat is spending immobile. Similar to what we've seen before, our court or stressed rats are spending significantly more time immobile. But what was remarkable was that rodents that received only a single intrahippocampal Relan infusion less than 24 hours before we assess their depression-like behavior, well, their depression-like behavior is completely gone. They are spending equivalent times immobile as our unstressed or vehicle-treated rats, which is remarkable. If we look at struggling, we see a similar pattern now where court rats are spending less time struggling, so they're more depressed. And our court rats that get a single Relan infusion are behaving like unstressed rats. And then with swimming, again, we see a single intrahippocampal Relan infusion is sufficient to eliminate depression-like behavior in these rodents. So this was remarkable because, as I mentioned, most antidepressants take 4 to 8 weeks to achieve therapeutic effects. I'm not going to be naive here and say Relan is the only compound that does this. If any of you have heard of ketamine, there's been a lot of interest in the depression literature in ketamine because ketamine is able to achieve antidepressant effects in as little as 24 hours as well. And then, as I mentioned, we ran them through the object location, so that hippocampal dependent memory test. And again, we saw that our stressed rats or court-treated rats were not learning this test. They were spending more time exploring the objects in the familiar orientation. And again, importantly, our stressed rats that were given a single Relan infusion learned the test and are performing at control levels. So we wanted to kind of figure out a mechanism. So how is Relan achieving these fast-acting antidepressant effects in our stressed rats? Well, as I mentioned, we had a separate cohort of rats that were given Relan on day 21. And then shortly before the forced swim test, they were given a single infusion of CNQX. CNQX is a glutamate receptor antagonist, specifically the AMPA receptor. And now what we see is that our court rats that were given Relan and CNQX, now they are not performing at, they are now acting like depressed rats again. So the antidepressant effect is gone. So that indicates that the fast-acting antidepressant effects of Relan require glutamate and specifically AMPA receptor activation, because when you block this, antidepressant effects go away. And we also wanted to look at if Relan is increasing synaptic plasticity. And so synaptic plasticity are just functions undertaken by neurons in the brain that enable basically learning and memory. And so we looked at mTOR and pTOR. mTOR stands for mammalian target of rapamycin, and it's been found that mTOR is upregulated by fast-acting antidepressants like ketamine. And it's also involved in neurogenesis and synaptogenesis. And it's been shown that mTOR signaling is decreased in depressed patients. And what we saw is that Relan is increasing mTOR expression and pTOR expression. So it's showing that Relan is also increasing synaptic plasticity in our stressed rats. So yeah, since I left the lab, that was kind of my last big project I did in my PhD. And so obviously there was a lot of interest in kind of furthering this work. So as I mentioned, we performed brain surgery on the rats to be able to infuse Relan into the hippocampus. That's not a very clinically relevant paradigm. As someone who's had brain surgery, I don't think a lot of depressed patients would want to sign up for that. So what they did was they just gave peripheral subcutaneous injections of Relan. And what they found is that antidepressant effects remain, even fast-acting, when you give a peripheral injection of Relan. They've also done some work where they showed that when you give ketamine to depressed rats, that increases hippocampal Relan expression. And they've also shown that Relan seems to have similar effects in terms of behavioral, electrophysiologically, and molecular, and a similar effect to ketamine. So some pretty exciting work. Because as I mentioned about ketamine, there's been a lot of interest in it. It's a life-saving treatment for people who are having treatment-resistant depression in particular, but it's not without its risks. So it is a dissociative anesthetic. It's a hallucinogen. So when patients get a ketamine infusion, they have to go into the clinic. They're monitored for hours. And there's also things like abuse potential. But there has been some work looking at like downstream byproducts of ketamine, as well as doing things like nasal sprays of ketamine. And so, yeah, the field is far from settled. I'm going to switch gears now and talk about Alzheimer's disease, which was the focus of my postdoctoral work. Alzheimer's disease is the most common cause of dementia. Broadly speaking, there's two types. The first is familial. Familial Alzheimer's disease refers to the inherited form of the disease. It is very aggressive. It has an early onset, typically in the 4th to 5th decade of life. The other type is sporadic. So sporadic is there are some genetic components to this form, but it's much more driven by environmental factors. It's less aggressive, and it also has a later onset, so typically the 6th to 7th to 8th decade of life. In terms of symptomology, what everyone thinks of as in Alzheimer's disease is memory loss. So there's profound episodic memory loss. There's personality changes, so patients frequently can become more aggressive. There's neuropsychiatric changes, so things like depression, anxiety are highly comorbid, and confusion and disorientation. Speaking at the neurobiological level, it's really characterized by two things: amyloid beta. So amyloid beta, it forms these plaques in the brain that can be picked up on when you do say when you do a postmortem investigation of the brain. So these are highly spread out throughout the brain, and they kind of deposit in this ordered fashion, which I'm not going to touch into. And basically, the takeaway is that amyloid beta is highly neurotoxic. It kills brain cells. The second thing is tau. So tau forms these hyperphosphorylated tangles throughout the brain. Again, they are highly neurotoxic, and they also spread in a pretty well-defined manner. So I want to switch gears a little bit and talk about glutamate. So glutamate is the brain's primary excitatory neurotransmitter and is heavily implicated in functions including long-term potentiation or the cellular underpinnings of learning and memory. So there are glutamate receptors in the brain that when glutamate is released binds to. So there's ionotropic. So ionotropic glutamate receptors are essential for synaptic transmission. And we can talk about AMPA receptors, kainate, and NMDA. And there's also metabotropic glutamate receptors, but that's beyond the scope of what I'm talking about today. So paradoxically, while glutamate is critical for the cellular functions underpinning learning and memory, too much glutamate is actually neurotoxic. Therefore, there has to be tight controls in place to ensure that glutamate is rapidly removed once it's released. And this largely comes in the form of glutamate transporters. So glutamate transporters are predominantly expressed on astrocytes. Astrocytes are a type of glial cell, and they surround synapses. And so, yeah, basically, once glutamate is released into the synaptic cleft from a presynaptic neuron and then went into a postsynaptic terminal, it's rapidly taken up by these glutamate transporters. The main glutamate transporter is EAAT2. I hate saying that though, so I'm going to refer to it as GLT1 because that's also what it's referred to as. So GLT1 is responsible for about 90% of all glutamate uptake in the brain. As you can see, you also have some other glutamate transporters, and these are also expressed on astrocytes. And you have small numbers of glutamate transporters expressed on the presynaptic terminal and postsynaptic terminal. So there's three scenarios I want to draw your attention to. This first one that I'm showing is a scenario of optimal glutamate uptake. So you have large numbers of glutamate transporters expressed on astrocytes, and when glutamate is released in the synaptic cleft, it's rapidly cleared, but there's enough to go on to activate other glutamate receptors, and you don't really have any spillover of glutamate into the extrasynaptic space where it would activate NMDARs, which can be neurotoxic. The second scenario here is when you have too little glutamate uptake. So here you can see there's not very many glutamate transporters expressed on astrocytes. You have glutamate that's being released in the synaptic cleft, but it's not being taken up fast enough. So now you have glutamate spillover into the extrasynaptic space where it binds to these NMDAR's glutamate receptors, which again can be neurotoxic. The third scenario is when you have too much glutamate uptake. So here you can see these transporters are enriched with glutamate transporters, and glutamate is being taken up so fast that it's not able to go and bind to the postsynaptic membrane where it can act on things like mGluRs, which are essential for synaptic transmission. So yeah, this would be a case of too much glutamate uptake. So there's kind of this Goldilocks zone where you want just enough glutamate uptake so that it's cleared from the synaptic cleft fast enough, but it also has enough time to go and bind to receptors that promote functions of synaptic plasticity. So about 20 years ago, there was a paper that was published that showed that the distribution of these astrocytic glutamate transporters is actually asymmetrical around hippocampal synapses. And what I mean by this is that there is up to 4 times greater astrocytic coverage of the postsynapse than the presynapse. And this paper was published 20 years ago, as I mentioned, and even till recently, there was little understanding of the functional consequences of this asymmetry. So I'm going to talk about glutamate dysregulation in Alzheimer's disease. So there's been a number of studies that have looked at human GLT1 postmortem expression, and the results here have been a bit mixed. There's been some papers that have shown GLT1 reductions in the hippocampus and prefrontal cortex of depressed patients postmortem, but there's also been other studies that have shown no changes in GLT1 postmortem. And some of the explanations that have been offered to account for this is that when you're looking at human postmortem work, you have to keep into account the interval between when the patient died and when the brain was extracted. So if you think of, say, cases of suicide, it can be sometimes it can be hours to a day to when the brain is actually extracted. And so as soon as death occurs, there's mass amounts of cell death going on in the brain. So you have to consider that when you're seeing these studies that are showing no effect, it could be just because of this extensive cell death that occurs. The rodent GLT1 expression literature is much more straightforward. There's been a number of studies that have shown GLT1 reductions across the hippocampus and prefrontal cortex in Alzheimer's disease mice. And we have to also talk about the role that amyloid beta plays. So there was this really interesting paper that showed that when you look at glutamate clearance in the Alzheimer's disease hippocampus, if you look at clearance that's, say, quite far away from these amyloid beta plaques, glutamate clearance appears normal. But if you go close to these amyloid beta plaques and you look at glutamate clearance, well, now you see glutamate clearance is significantly slowed down. So amyloid beta is playing a significant role in glutamate clearance in Alzheimer's disease for sure. The Alzheimer's disease brain is also characterized by reactive astrocytes and microglia. So in Alzheimer's disease, you'll see astrocytes and microglia change their state to a reactive state. And these reactive stages significantly impair glutamate uptake. They slow it down. And also, when you consider glutamate in Alzheimer's disease, a large focus of the research has focused exclusively on the postsynapse. So what are the contributions of the presynapse? Well, that's been examined a lot less. So this brings me to the main publication of my postdoctoral fellowship, which was looking at glutamate dynamics in a mouse model of Alzheimer's disease. So what we did is we worked with Alzheimer's disease mice, and specifically the 3xTG model. And what we did is we performed, again, brain surgery. So we would drill a hole down into the hippocampus, or not down to the hippocampus. We would drill a hole in the skull, and then we would infuse a needle down to the hippocampus. And what we would do is we would infuse this compound called I-glu-sniffer, which stands for intensity-based glutamate sensing fluorescent reporter. So basically what I-glu-sniffer is it's a genetically encoded fluorescent sensor that lights up when glutamate is released in the brain. And it's a virus, so it's taken up by neurons. And what we did is we were able to inject either into the CA3 region of the hippocampus. So this is a presynaptic region. The cell bodies in CA3 project over to CA1. So this is a presynaptic connection. Or we injected specifically into the CA1. So here you have the terminals from CA3. They terminate on the cell bodies in CA3 and the CA1. So this is a postsynapse. And then we're able to look at glutamate clearance in the two regions. So you're probably wondering, well, what does this look like? So we use two-photon microscopy, it's called. It's a very sophisticated means of looking at glutamate clearance in the brain. It was very stressful to use. My supervisor's microscope was like $1 million. So I had a lot of stress dreams about breaking the microscope all the time. I don't have them anymore, thankfully. But yes. So here what you're seeing is a dendrite on a postsynapse. So you see that lighting up? That is we deliver a pulse of electrical stimulation, and the lighting up is glutamate being released. And then you see it, the intensity goes down. So that's it being cleared. So that's what it looks like. So the first thing we did is we wanted to test that paper I brought up, which showed that there is a asymmetry of hippocampal synapses in terms of this glutamate transporter coverage. So what we did is we delivered 5 hertz of electrical stimulation to either the postsynapse, so these hippocampal dendrites, or presynaptic boutons. And we delivered this electrical stimulation in the absence or presence of something called DHK, which is a GLT1 antagonist. So it's blocking GLT1 in that area, or it's lowering the levels of GLT1. Our rationale for this is that as the postsynapse, as I mentioned, is enriched in these astrocytic glutamate transporters, we want to try and bring GLT1 levels down there so that we can kind of bring them down close to the level of the presynapse. And so when we did that, what we found is that glutamate clearance is slowed to a greater extent at the presynapse than the postsynapse. And so what this shows is that indeed the presynapse is indeed more vulnerable, and it has slowed glutamate clearance dynamics than does the postsynapse. And I want to point out that this was done in healthy, non-disease C57 mice. So in the healthy brain, what we're showing is that indeed glutamate clearance is slower at the presynapse than the postsynapse of the hippocampus. So now what we did is we used our 3-XTGs, so our Alzheimer's disease and just healthy wild type mice. And again, we put iglucinifer into either the postsynapse, so the CA1, or the presynapse, the CA3. And we did 5 pulses of stimulation or 100 pulses of stimulation. And when we look at the postsynapse, what we see is that at these short 5 pulses of stimulation, glutamate clearance is not differing between the groups. When we increase it up to 100 pulses, so a much greater challenge, again, we see that glutamate clearance is the same between the groups. There's no difference in glutamate clearance at the postsynapse between Alzheimer's disease and non-Alzheimer's disease mice. These are just some representative pictures showing glutamate clearance between the two groups. You can see they look pretty similar. But now when we focus on glutamate clearance at the presynapse, so at the CA3 region of the hippocampus, when we look at 5 pulses of stimulation, both groups are the same. But when we increase it up to 100 pulses, now what you're seeing is that shown here in orange, our Alzheimer's disease mice are significantly slowed at clearing glutamate than our wild type mice. So Alzheimer's disease mice are significantly impaired at clearing glutamate at the presynapse. Again, this is a representative picture showing you our wild type and our Alzheimer's disease mice, and you can really see the difference between the two groups here, with the 3-XTG mice being a lot slower at clearing glutamate. So these first experiments were done in 6-month-old mice, which obviously that sounds young, but in mice that roughly corresponds to almost middle age. So we want to repeat these experiments in older mice. So we used 1-year-old mice now. So these are pretty old mice. Oops, sorry. Yeah, okay. So we looked at glutamate clearance in the postsynapse. Again, we used our 5 pulses of stimulation. You can see there's no significant group differences here. When we go to 100 pulses, again, no differences. So even in old age, the postsynapse is still functioning quite well in Alzheimer's disease mice at clearing glutamate. Again, representative pictures showing you pretty similar clearance rates between the two groups. And when we look at the presynapse, we see at 5 pulses, there's not a huge difference. When we jump up to 100 pulses, again, we're seeing an even more exaggerated clearance deficit in our Alzheimer's disease mice, where they're significantly impaired at clearing glutamate from the presynapse. So these are just some representative pictures again. So the next thing we wanted to do is we wanted to see if we increase GLT1 expression, do we restore glutamate dynamics? So we repeated our imaging experiments in a separate cohort of mice that were now given IP, so intraperitoneal in the gut, injections of ceftriaxone. Ceftriaxone is actually an antibiotic, but it's heavily used in neuroscience because it's in a GLT1 agonist. So what we find here is that we replicated our earlier findings, where at the presynapse, our Alzheimer's disease mice are significantly slowed at clearing glutamate. But now, in our Alzheimer's disease mice that were treated with ceftriaxone, well, they're behaving like non-Alzheimer's disease mice, and they're not impaired at clearing glutamate at the presynapse. So what this suggests is that GLT1 is dysfunctional in the Alzheimer's disease hippocampus. These are just some representative pictures here showing you clearance rates between the two groups, and you can really appreciate how you see at the top we have just wild type mice. The second panel is our Alzheimer's disease mice, and when you look at the bottom, which is our Alzheimer's disease mice treated with ceftriaxone, you can see the dynamics are looking a lot more like our wild type or healthy mice. So interestingly, when repeating our previous experiments, looking at using ceftriaxone, we now focused on the postsynapse. And what we found is that ceftriaxone altered glutamate dynamics in this microenvironment in a pretty unexpected way. So at 5 pulses, the groups are not different. But when we go to 100 pulses, now what we're seeing is that glutamate clearance is actually slowed in our Alzheimer's disease mice at the postsynapse that are treated with ceftriaxone. So we were a bit perplexed by it, but notably it didn't reach statistical significance, thankfully. So we were a bit perplexed by this finding. But one suggestion was that it might be a nanoscale misalignment of glial membranes, where the previously reported glial asymmetry favoring postsynaptic protection is actually exaggerated even further in 3XT in our Alzheimer's disease mice. But yeah, this is a pretty unexpected finding, and we did have a hard time explaining it. So again, these are just kind of some representative pictures. So now I want to get to, well, what are the consequences of this? So we've demonstrated that glutamate clearance is slowed at the presynapse of Alzheimer's disease mice, but who the hell cares? What's it mean? So we wanted to understand the impacts this clearance asymmetry has on essential functions of neurons and synapses, specifically short-term plasticity in the form of post-tetanic potentiation, or PTP, which is characterized by a temporary facilitation of presynaptic release. So we inserted electrodes into the hippocampus of brain slices obtained from Alzheimer's disease and wild-type healthy mice. And so what we found is that post-tetanic potentiation was decreased in Alzheimer's disease mice. So what this means is that their synapses have a reduced release probability. And so this matters because presynaptic glutamate receptors exist in the hippocampus, and when glutamate binds to them, they decrease glutamate release. And so over time, this opposes synaptic plasticity and weakens the synapse. And over time, this could lead to synapse silencing, a key feature of Alzheimer's disease. So what we're seeing is at the presynapse, glutamate clearance is slowed. There's more glutamate hanging around. And now this is going and activating these presynaptic glutamate autoreceptors that limit the glutamate release. And over time, this is going to weaken the synapse and they might become silenced over time. And so we finally asked if we increase GLT1 expression, would this reverse the synaptic plasticity deficit that we saw in our Alzheimer's disease mice? So indeed, we found that if we increase GLT1 expression via ceftriaxone, we now bring post-tetanic potentiation to healthy wild-type levels, and their synapses exhibited an increased release probability. So that was it. Yeah, happy to answer any questions.